What is the genetic material? What is the gene? Gene is the information the parent transfers to the offsprings, and this information should be stored in just a single cell, sperm or egg cells. So that's why the genetic information should be highly condensed form of information. Then the next question is, what is the storage material for this genetic information? For a long time, there were two candidates. One is the DNA, and the other one is the proteins. Among these two options, people believed maybe the protein would be the genetic information. That's because protein is the much more complicated and also has a more diverse other characters comparing to simple and boring DNA molecules. To find the answer, if that is your problem, how will you design your experiment? Well, here is the two scientists designed this experiment, and their name is Hersh and Chase. For this experiment, they chose bacteriophage. Bacteriophage is kind of a virus, and this virus use bacteria as their host. Bacteriophage has a protein coat, and also they have a DNA inside. So they have both protein and DNA in their form, and then this has been used for the experiment. Bacteriophage use bacteria for their host. So they attach to the surface of a bacteria, and somehow, after some time, they multiply and make more copies of viruses. So utilizing this system, how could you figure out whether DNA or protein is genetic information material? When the virus attaches on the surface of bacteria, they inject the DNA material into the bacteria, and the protein coat side remain on the surface of a bacteria. Now let's look at the details of experimental design of Hershey and Chase. They decided to label proteins. Protein has uniquely sulfur element, and common sulfur is a sulfur 32. That's a stable isotope. They chose the sulfur 35, which is radioactive, and then they labeled the virus particle with the sulfur, then they can label the protein selectively. In another case, they utilized phosphorus to label the virus. Phosphorus only exists in DNA, not in protein, and the stable isotope is a P31, and they utilize the P32, the radioactive version. So now they have two labeled versions of a virus. The first experiment was done with a protein-labeled virus. First of all, the virus attaches on the surface of a bacteria, and they inject their DNA into bacterial the inside. Then many copies of a new virus are formed. Then after vigorous shaking, the surface attached original virus was removed. Then the radioactivity was measured. Very interestingly, the newly synthesized virus particles, they did not include any radioactivity. This experimental result implies that the protein from original virus was not transferred to new virus particles. So that means protein is not genetic material. The second experiment was done with a DNA labeled virus. Here the virus attaches on the surface of a bacteria, and they inject the DNA into bacteria. After some time, they formed new virus particles. After vigorous shaking to remove the original virus particles, they measured radioactivity of newly synthesized virus particles. And they found that the new virus particles, they are radioactive. So this result shows that the original DNA was transferred to new virus particles. That implies that the DNA is the genetic information material. With this experiment, now it's clearly demonstrated that the genetic material is DNA, not protein. It's a beautiful experiment, isn't it?